All right, hi everyone. So this is a retake of the lecture that I gave earlier in the week, and I'm going to call this the director's cut. I also neglected to go through this slide saying that it's been 10 years since I started teaching here in HMB, and I've been teaching HMB300 for about 9 years. So very excited. I hope that you're also excited for this semester. Sorry that there were so many technical glitches, and I hope that every class this semester we're going to have a really enjoyable time of getting to know each other, to make some connections, and that they become un-LTD'd. So no depression, all strengthening. So today we are going to go through the director's cut version of the lecture that I gave earlier this week, and this is again sort of an extended version, things that we've learned from the particular lecture. I won't go through the notonics because that's separate and I don't want to go through their slides. If you do have any issues with notonic, there is a contact information for you, support@notonic.com. But today, I really wanted to go through the bias of neuroscience. And neuroscience is incredibly biased. So just think of the name, neuroscience. It actually tells you that neurons are the focus of what we study within our discipline. And maybe that's the wrong thing for us to be doing. And I'm going to try to take you through why I feel that there is a neuroscience bias and why this has to change and how it is already changing. And along the way, we're going to be looking at what we call unicorns. And these unicorns, do they really exist? Are we not sure? If we're not sure, why are we not sure that these unicorns exist? And that's going to be sort of the sub-theme throughout the entire course and throughout this entire semester. And I hope you're going to have an amazing time. This is a wellness-focused course, by the way, so at any point in time, if you need to chat with me, if you need to have extra material, if you need extra help, please let one of us know or myself know, and we'll be happy to try to make accommodations. I really, outside of the content that I want to talk about today, that's the one thing that I want you to get across, that it is a wellness-focused course. So, before we begin, one of the announcements that I made in class was that the syllabus quiz. I had asked that if most of the class had actually finished the syllabus quiz before the first lecture, which was back on September the 11th, that I would automatically round at the end of the semester any grades that were sort of on the GPA bubble. So if you were sort of at a B and you were on the cusp between a B and a B+, that if enough students actually did the syllabus course quiz in advance, that I would actually go ahead and round up. And I can tell you that, yay, just like Snoopy, everyone did in fact do it. We met the threshold. In fact, we smashed the threshold. About 80% of everyone registered in the course at the time of the syllabus quiz actually was able to finish it. So I was incredibly proud of all of you. So my objective for you in this course is to really make sure that you understand the importance of neuroscience, but also that you're taking good care of yourselves. And I do want you to feel like you're engaged and that you're connected, and that's sort of the focus of the next few slides. And you can go ahead and skip through them if you don't need any help. You feel like you know all of the administrative details, and then I'll tell you when you should come back and pay some more attention. So what we're going to go through over the next 5 to 10 minutes is sort of a summary of everything that I gave on Wednesday about some of the questions, a little bit of feedback that we received in class, some of the different topics that we were going to cover in the course, and starting to introduce to you the fact that this gut that we're going to be looking at today, a little bit later on, is actually impacting the brain a lot more than we thought. And again, this is sort of the neuroscience bias that we're going to cover a little bit later on. So I am going to also suggest to you how to study, how to use today's lecture in fact, and how to study throughout the semester. And that's important to me, that you're learning how to study if you're not already comfortable with that. We will also be providing you with occasional workshops on these assignments. We're also going to give you assignment guidelines. We're going to give you different things to help you succeed wherever possible in this course. And of course, I'm going to tell you that we are biased, incredibly biased within neuroscience, and this is a big problem. And this has actually led to a lot of setbacks that I'll talk to you about in a few slides. Also, should you brush your teeth? Well, of course you should, but maybe not for the reasons that you're thinking. And at the beginning of the next lecture, I will actually go over the oral microbiome and its impact on your brain as well. So lots of things to cover. Hope you're excited. So let's go ahead and dive right in. So I don't really have anything much to disclose to you. That neuroscience textbook is completely free. I'm hoping that some of you at the end of the course will also help to revise and write the next edition of this neuroscience textbook. And I really want you to love neuroscience. That's incredibly important. I'm hoping that some of you love neuroscience so much that you go on to think about a career in research in neuroscience. And hopefully, if you win a Nobel Prize, you will think back to your HMB300 prof, invite me along to when you go pick up your medal in Sweden. So I do, in fact, have an ulterior motive. One of the things that I would like you to all consider doing is to join a recognized study group. And for those of you who have joined this course, these recognized study groups, we had someone come in and talk about them, and I've also put up the links on the discussions page. Again, no direct benefit to me. I have no association with them, although I will be dropping into recognized study groups to help out wherever I can if you invite me and if you tell me like where your recognized study group is meeting. I really want to emphasize that we want you to do well in this particular course. So this is one of the polls that we actually had in class, and we wanted to know, because of Notonics being there, and Notonics is something that is free for all of you. We're going to use it as participation. We haven't started yet, so no panicking, no stress. And we just wanted to know like where everyone was at. How do you take notes in class? And this was the results of the polls that we actually took. And it seems that most of you are actually either annotating the PDFs directly, or you are doing Microsoft OneNote, or some of you have mentioned in class that you're doing none of the above, and in fact one of the things that you're doing is just using good old-fashioned pen and paper, which again, incredible if you're thinking about it. So that said, we did do that poll really quickly. And these are sort of the office hours I had mentioned in class as well, that generally speaking, I'm going to be available to everyone in class, and I hope I can meet everyone in class. It's important to me to get to know you. I have a lot of social phobia, I have a lot of social anxiety, but I am happy, and I'm incredibly happy to meet with students. And even though I may be really awkward and shy, and you might feel that you're awkward and shy, let's meet together and let's try to help each other out with this. So again, Thursdays 3 to 4, Fridays 1 to 2. There is actually a video online on where to find my office. You're free to drop in, chat about whatever's on your mind, about coursework, about assignments, etc., or just talking about life or what you're going through. I'm happy. The door is always open for you. And again, the course policy is, wherever possible, we're going to use the discussion board on Quirkus, and I've already set it up, and there's been lots of great questions, so thank you for that. I really do want to emphasize, you're going to be excellent in this course, and our job is to help you to reach that goal. And I really want to emphasize that in this course, starting next week, so on September the 18th, that when we start meeting in our amazing room in My Hall 150, that we are going to be really working toward making sure that you feel comfortable with all of the different assignments and assessments, that you're coming together as a class, and that we are there to help you. And the 4 questions that we'll start with in the next class, you'll see right away that you will need to be able to do some problem solving, that you're going to work together in groups, and that we're going to have some fun with it along the way as well. If you need to contact me, you can use either my Gmail account, or if you have an administrative issue, something's gone wrong, you need some help, and you need some extra resources, more time, you need to reach a TA and you can't do that, you need to talk to a PA or one of the peer assistants, then try to reach me at my U Toronto. If you put HMB300 in the subject line, I will immediately respond as soon as I read it or open it. Okay, so please try to connect with me wherever possible. So here we are at this slide, and the way that I presented this earlier is really to talk about what these different things represent. So microglial cells just kind of show up randomly whenever they need to in the brain, and so these microglial cells are really not considered to be part of the brain. Embryologically, these microglial cells actually come from a very different source. They're actually not brain cells. They actually come from a different embryological origin. And so I don't want you to be like microglia, not just showing up randomly and only for term tasks. Wherever possible, engage, whether it's in person or online. Please don't just randomly show up here and there. I don't want you to be astrocytes either. Astrocytes are important, and we're going to talk about astrocytes an awful lot in this course, but astrocytes, again, they play more of a support role. They kind of hang out. They only become reactive when something goes wrong. And these reactive astrocytes are not what I want you to be. I want you to be more like these fully connected neurons, where you're strengthening your connections with other individuals in this course. And I also want you to strengthen your connections with me. And again, one of the ways that you can tell if you are strengthening those connections, this is a very common phrase here at U of T, to fake it till you make it. And that's totally wrong. If things are not going well for you, I need to know as soon as possible. For me to help you, I need to know that there's something wrong. And I'm going to give you a story of what happened to me when in undergrad. I used to fake it, and I didn't make it. I didn't make it until about my third year. And most of the time when my friends were asking how things were going, I would tell them everything's good, my classes are going well. But in fact, I wasn't doing well, and I was always feeling very frustrated. And I kind of wish, looking back, that I had a lot of help from my profs, from my mentors, peer assistants, which weren't available at the time, and I don't want you to fall into the same trap. So please connect with me whenever you possibly can. I know it's uncomfortable, but I promise that we'll figure out a way to connect with you. And I don't want you to just survive. I want you to thrive in this course, and that's really important for us. So I want you to be the best neuroscience student that I can possibly make you to be. We are going to have lectures throughout this course. We're also going to have active learning in this course, and we want you to lead the way in terms of learning. I want to connect you within this class to other individuals, whether it's online. So I know some of you are listening to this online and may only be able to listen to this online, and that's okay. You're still able to connect, and that's more important. You don't have to be in class to be able to connect. I'm not saying that if you are online that you're a microglial cell. I'm just saying that there are other ways that I can help you with to connect and feel part of this class, whether it's through social media, whether it's through discussion boards, but I do want you to feel connected, that you're feeling comfortable, that even if you're not in class, that you feel like you know people in class, as well as within the program. I also wanted to tell you that neuroscience changes incredibly quickly. There were so many changes in neuroscience and so many groundbreaking papers that I want to present to you throughout this course, and that's one of the big things that I want to get across. I also want to help you refine some of the skills that you're going to need throughout the rest of your career, like writing, how you're going to present some of the different data in the different papers that you're reading, how you're analyzing the different data in the papers that you're reading. All of these are important. You're going to need them no matter where you go, whether it's in neuroscience, whether it's in biology, whether it's in law, whether it's in medicine, or whether it's in another professional program or in the workplace. These are all important. So I want to, again, really encourage you to go over the course syllabus. I want you to make sure that you are understanding like what your requirements are, and I will always be updating the Quirkus site for you to make sure that you know what you need to be able to do every single week. We will provide, and unfortunately every last thing broke down, didn't work, terrible. We will always provide you with lecture recordings. We will always provide you with different resources, whatever they are, and we will always be available for help, either online or in person. So if you have any questions about unpacking the course and what you need to do, we will give you assignment guidelines. We will make sure that you know the rubrics in advance. We will be holding extra office hours, workshops, whatever we can do to help you succeed in this course. So one of the reasons why we're going to use this particular classroom in my hall is that this paper that came out earlier in PNAS, which is a fantastic, highly ranked journal, and maybe some of you might find your paper that you're going to do a lit review on within PNAS, is that if you have a fantastic lecturer, I'm not in that category unfortunately, but if you had a fantastic lecturer, you might feel like you're learning something, you might feel like you're gaining a lot from that classroom, but when it comes to the actual test and the actual evaluation, and you probably have had that experience, that you're actually not going to perform as well as if you, or unless you had actually engaged in what we call active learning. So thinking through a problem together in groups is better for your learning. You'll retain it longer, according to this paper, and actually this is true. We found this in our own research. If you're actively engaged in the classroom, which is our responsibility, then you'll be able to remember things better, problem solve better, and do better on all of your evaluations. And that's our goal. So next week, we're actually going to have some active learning exercises during the first 10 to 15 minutes of class. So exciting times ahead, folks, exciting times. So I'm also going to be providing you with an end of the week to-do list of suggestions, and I'm hoping that you will go through them. This week I will be posting it on Friday afternoon. So later on today, if you're hearing this today, but you should go through them over the weekend. There isn't going to be a lot. I will also be providing you with a study guide, and it's just a list of suggestions. You can study however you want, but I think that this might be helpful for all of you. So I'm going to be working backwards in this course. So we're going to be targeting all of our active learning exercises to get you ready for the term test, for the final exam, for the assignments, etc. So it's going to be exciting. We're going to jump right in starting next week. And I'm also going to take the unusual step, which I've never done for any class before. So you guys have made a huge impact on me already. That's starting next week. Or starting very soon, I'm going to be posting previous midterms as well as final exams for you so that we can actually start this discussion about getting you ready for all of these different types of assessments. Again, we have examples for you. We will have rubrics. We will have assignment guidelines well in advance of the October due dates for the visual summary as well as the final written assignment, so you can go in and take a look. The peer mentors that are available for you, or the peer assistants, many of them have had their articles published, and you can talk to them as well. So lots of examples, lots of things for you to refer to. And again, Jessica is one of the peer assistants this year that you can go and talk to directly about how long it took, what were some of the things that she would wish that she could go back and revisit if she were to have to do this again. So hopefully, because we're providing you with all these resources, you will feel like you are going to be very successful without too many worries. And again, if you do have any worries or concerns, we're going to be helping you. We're going to also give you more details about this visual summary coming up. And this visual summary, this was actually created using a free program called BioRender. If you have artistic skills, you don't need to use BioRender. I'm going to also do the assignment with all of you. So I'm going to pick a paper. I'm going to post that paper, and then I will not have read it. It will be a brand new paper. And then I'm going to go through using the guidelines, and you'll see sort of my reaction video as I go through and start doing this visual summary myself. And you'll see how long it takes. You'll see like what sort of struggles I have or what I throw at the computer screen when things don't work. So I also wanted to tell you a little bit about why we're doing all these things. So you might think, well, can we just not have term tests and final exams? One of the ultimate goals that I mentioned to you at the outset is that I want to stay in contact with you. I'm hoping that you'll go on to successful careers wherever you go. But wherever you are going, you're going to need a little help getting there. And so part of the goal for me to have these assignments, to work with our TAs and peer assistants, is to really see that you are cooperating in groups within these active learning exercises, that you can communicate in written form and with me when you're showing up to the office hours, that you can do this self-directed learning because we will provide you with weekly study guides, and that you also have really good critical thinking ability. It's not just about memorizing things. This course is much more specific to problem solving. And I know that sounds really scary, but with these active learning exercises coming up next week, you'll see that it really isn't scary at all. And please just feel comfortable. Feel like you're going to be able to do a great job on all of these, and that when you ask me for an evaluation or a reference letter, that you will have like no doubt that I'm going to be able to provide you with a strong one. So some of the suggestions that I have for you for studying, there are going to be slides that have bullet points. There are going to and only bullet points. There are going to be slides that have both bullet points as well as figures and diagrams. And as you'll see very shortly, there are also going to be some slides that only have figures as well as diagrams. No bullet points, no writing, or very minimal writing. And when you see these, you're going to have to take some more notes down, and you'll get into the habit of doing so, and I'm hoping that you'll use Notonic or other programs, and that you'll get into the habit of being able to take down these different notes. So indicate on your notes, and you can do this through Notonic, or you can just jot down a note if you're doing it by hand, what concepts you don't understand. Review those as soon as possible after lecture. Whether they're slides that have bullet points that made sense to you while you're sitting there in lecture and they don't make any sense like half an hour later, or they don't make sense to you like a day later, then you should come in and see me. And then we're going to also start taking you through questions that are scenario and application based. So don't just memorize. You need to do a little bit of that, but also start thinking about how all these different ideas connect together. And we are going to be providing you with resources to help you study. So these recognized study groups, when these study guides are available to you, they're a great way for you to review, compare answers, talk to each other, explain these different slides that you need to know to each other, and to think ahead, like what should we really get out of this particular slide. It's going to be fun, it's going to be exciting, and I hope that you're also going to be loving HMB300 as much as I loved the first lecture. So I'm going to tell you the one thing that students often have a really hard time with. So one issue is that if you're showing up to every single class and you're taking down notes, you might feel like you're actually accomplishing a lot, but that was me. That was me in undergrad. And in undergrad, I would really just focus on showing up to lecture, taking down notes. I didn't really study for the term test, or I didn't really study and prepare for the assignments. And we want you to be ready, to be more prepared, not just showing up to class, not just listening to the lecture, but also being well prepared in advance for the types of questions on the term test, and maybe down the road after this course to use the same techniques that you've learned here in your other courses. So we want to help you learn how to study a little bit more efficiently. And one last thing, time management for me as an undergrad was like horrific. I used to like procrastinate like you wouldn't believe. And one of the things that was really wrong about that was that I used to leave things until the day of the term test. Like I would literally have about 8 or 9 lectures in physiology that I kind of had to review. I thought I was well prepared because I'd gone to every class, but then when I went back and started reviewing the day of the term test, you can imagine it wasn't very fun. And it's a lot like eating. So eating is a great sort of activity. I love eating almost anything, but imagine instead of eating like 1 meal or 2 meals a day on a regular basis, like you left all of that and you were given like 1 hour to finish like everything that you would normally have eaten throughout the course of like 6 weeks or 4 weeks, all on one day within like 1 hour. It wouldn't be a very enjoyable experience, I don't think, and I'm hoping that you don't take that approach. Don't be like me, do better. And so I'm hoping that you will not have this wrong focus. All right, we're almost there, folks. So here is where we had a BS checker. So this is a blind spot check. I like to think that I tried to prepare as best as I could, and I put up this slide, and I don't want you to feel like, hey, I feel all prepared, it's going to be great, and then suddenly everything is taken away from you. And when I put this slide up, there was time available in class for students to respond, and some of the suggestions that came out of this blind spot check were to have suggestions for loading the notes earlier rather than 24 hours. Could I post them at least 48 hours in advance? And I will certainly try my best to do so. There was also an individual who came in to actually talk about recognized study groups and how to register for them. I have now put this on Quirkus for you, and one of the individuals from the Sydney Smith Commons actually told us about the Meet to Complete program. Another option was to engage in study hubs, and these study hubs you can ask either Rena or Bakiza, who are our official ambassadors. You can also talk to the TAs or the peer assistants as well, and we introduced all of them in class. And I'm hoping that that was everything. Again, I should have another blind spot check for this blind spot check, but I don't. So I think that that's what all I remember from class in this director's cut, and I think that's it for the administrative stuff. So if you want to take a pause here, or if you're just joining us now after this pause, and now we're just going to jump right into what you need to know for the very first term test, some of the content, and everything else. And the first slide is again, I wanted to just know, where are you coming from? Why are you interested in neuroscience? And that poll question, it turns out, well, neuroscience is cool. I'm hoping it's cool. And I'm hoping that I'm going to try to convince you that it's cooler than you thought. And if you have no idea, I'm going to try to convince you that it is maybe cooler than you thought. So we're going to jump into that next week. So hold on tight, folks. We're going to have like a wild ride throughout this course. We're going to chase down some unicorns. We're going to capture some unicorns. And we're going to figure out why they're unicorns throughout this course. And for those of you in this category here, if you're thinking about Nobel Prizes, just don't forget your prof. Invite me along. I'd like to visit Sweden one day, and it'd be really cool to be a tag-along or a +1 at this ceremony. All right, folks, we're almost there. So why are you interested in neuroscience? Why neuroscience? Well, the analogy I gave on this particular slide, which of course you don't have to memorize, and it was just here to kind of get you excited. Like, I got very excited with this slide because when I was thinking about where we're at right now in neuroscience, where we're at is really the same place that astronomy was at back in the 1600s. And in the 1600s, where we had these amazing researchers, or we had these amazing astronomers, Tycho Brahe, Johannes Kepler, and Galileo Galilei, all of them were actually at the forefront of astronomy, and they were changing astronomy because astronomy had sort of the wrong focus. And the wrong focus was that the Earth was the center of the universe. Everything revolved around the Earth. It was there and held in place for a very, very long period of time. And these scientists came around and they actually found the scientific proof that, in fact, it was not the case at all. Now, keep in mind, they were surrounded by these giants in the field and these amazing individuals, incredibly smart individuals who just didn't know that everything was different. And so these important astronomical discoveries actually led to more and more powerful telescopes, and these telescopes helped to make more and more important discoveries that changed the focus of astronomy. Instead of looking at the Earth as the center of the universe, that we were just part of the universe. And in some ways, neuroscience is in exactly the same place. We talk about the brain, and we talk about diseases like neurodegeneration and Alzheimer's disease in the context that everything must originate within the brain. The problem is, over the last 70 years, because we've been so focused on the brain as the primary organ and the originator of neurodegenerative processes, that we've been blinded by the fact that it's just one of the many systems that could impact on the brain. And so we're really, right now, in 2019, in this incredibly exciting place, just like we were at the turn of the 1600s, we're in this really incredible place where we're seeing a shift, and a dynamic shift away from just thinking of the brain as the primary place, but we're also now thinking of the gut microbiome, and other types of very interesting phenomenon that impact the brain as well. And we'll see that some of these are unicorns. We think they exist. We're not completely sure. Maybe they're fictitious. Maybe they're mythical. Maybe things like meditation have a physiological impact. Maybe they don't. Maybe things like placebo are just simply unicorns that don't really exist. Or maybe there's a biological mechanism for both the placebo and the nocebo effect. And of course, exercise, a big issue in neuroscience right now. How does it work? Is it a unicorn, or is there a physiological, research-driven sort of way that we can measure the impact of exercise and its benefits on the brain? So this is where we're at right now, in this exciting period of time within neuroscience that leads to a brand new field and maybe a brand new sort of outlook on what the importance of neuroscience is. And so I'm going to introduce, as I did in class, and I didn't put this picture up, so if you're coming back for the director's cut, so this is added and it's kind of new, this is Dr. Rudolf Tanzi, a leader in the field of Alzheimer's research, and he has been for a very, very long period of time. The problem with Alzheimer's disease research is that people have known about Alzheimer's disease for well over 100 years. Well over 100 years. And so how many different types of therapeutic interventions do we have for Alzheimer's disease right now? Zero, none that have been proven to work. And this has actually caused a dramatic shift and a rethinking, a refocus. The brain may not be the center of our universe anymore, that if all of our traditional theories over the last 50 years, because I was involved in this research as part of the TANS Neuroscience Research Institute here at U of T. My research was also in Parkinson's, Alzheimer's disease, and it was all thought to occur within the brain, but maybe it isn't. And Rudolf Tanzi actually started to rethink this, and he's the director of neuroscience at Harvard University. And one of the things that he started to notice is that individuals that had Alzheimer's disease, when he examined the brains of these individuals, they also seemed to have high rates of infection where he could detect that they had a particular type of bacteria known as Borrelia. And this Borrelia Burgdorferi is a type of spirochete, so the spiral-shaped bacteria that he found in the brains of individuals that had Alzheimer's. And a lot of people, even though he was a well-respected researcher in Alzheimer's, when he made that correlation that maybe bacteria are actually the cause of things like Alzheimer's disease, people laughed at him, even though he was an incredibly well-respected individual within the field. And part of this, even though there's an association between bacterial infection and pathogenesis and degeneration in the brain, so Borrelia causes Lyme disease, and he sees a lot of this Borrelia in patients that also have Alzheimer's disease, people were laughing at him, saying that it can't possibly be true, or it's just correlational. In fact, maybe because they have Alzheimer's, they're actually opening up the blood-brain barrier and they're just more prone to infection. So it's not the cause, it's more of just an artifact of the actual process itself. But now we're kind of re-examining this. We know that different types of bacteria, whether they're in the oral microbiome, whether they're introduced in other ways, or whether they exist and can actually exist within the brain. Again, all brand new territory, and I've actually posted a paper on the discussion board about this. These are some of the mechanisms that we talked about in class. And some of you are asking about this. Well, these are Easter eggs. And if you know what an Easter egg is, this Easter egg is there for a reason. And again, think a little bit about what an Easter egg means on a director's cut on a video. So neurons actually have this molecule known as amyloid precursor protein, or APP. And this is the precursor version of the beta amyloid that we often think about, or amyloid beta that we often think about in terms of Alzheimer's. And we don't really know the mechanism by which amyloid precursor protein can actually be converted into this beta amyloid, but we know that as you accumulate more beta amyloid through mechanisms that we're not 100% sure about, even after 50 years of research, where over $10 billion of research money have been spent by countless labs across the world, we don't know how this beta amyloid actually gets processed. We don't really know all the details around how this causes the death of neurons, but we do know a couple of things, at least from this bacterial perspective. Bacteria, and here is a representation of bacteria, actually have proteases that can cleave amyloid precursor protein into this beta amyloid. Again, it doesn't explain how it actually causes the death and the accumulation, but it's also like really interesting. These pathogenic bacteria can also clump together, as you're seeing here, and they can actually form a biofilm, and these biofilms are resistant to immune attack, and these immune-resistant forms of bacteria can also combine with amyloid beta, which again, they might be contributing to in the first place, to actually cause the amyloid beta to be evasive or to evade the immune system attack. And again, we've tried different types of antibody therapeutics against beta amyloid. None of them have worked, and no one's been able to come up with a reasonable explanation of why. But this is sort of an alternate hypothesis that is gaining traction and gaining ideas. We also know that microglial cells, the immune cells, and remember we talked about microglial cells kind of coming in, coming out, staying there in the brain, but not really doing very much. These microglial cells, once we start this process, can be activated by the beta amyloid to actually cause the release of various pro-inflammatory cytokines, which in turn may cause the death of neurons. But all of this really results in having some kind of bacterial infection within the brain. And it's been thought for a very, very, very long period of time that the brain can never have bacteria within it, that it is a privileged space where bacteria cannot exist. And this dogma, this idea, just like the Earth is the center of the universe, this idea is being challenged in a number of different places right now. So again, exciting times, folks, incredibly exciting times. And it turns out that we then took a break, and we are going to start having some quizzes, so you can start doing these for your 5%. You don't have to do every single one of them, as I mentioned. Some of you have already done one of them for up to 1%, and we do have some participation marks on these quizzes, and I'm going to start these. You can do them all the way up until the midterm if you need that extra time. We will only take the highest mark. You can take them as many times as you want, and all of these quizzes will have exactly the same format. So again, no stress, folks. So excuse me on that. One of the things that I talked about on this particular slide was that in this course, you're also going to be hearing a little bit about my own personal journey, why I'm interested in these different types of topics. And one of the things that I mentioned in class, and I will mention this again later, and I'm not embarrassed to mention this, is that in my time within the university as an instructor, as your professor, I've actually experienced depression twice. So once was about 8 years ago. It was a mild form of depression. I really didn't understand what was going on. And then I had a much more serious bout that I'm going to go into some more detail later on, about 3 years ago. And this was a very, very severe form of depression. It was true depression. All the things that you would think about associating with depression, all of that happened to me. And one of the things that happened as I started to undergo both the early 8 years ago versus the one that happened 3 years ago, I really started changing my eating habits. I started to change what I liked doing. I started having various aspects of my metabolism being affected. And it turns out that even actresses like Gwyneth Paltrow, as they take on new roles and as they change their eating habits to gain weight for different roles, they will also undergo a number of these different mood disorder changes as well. And I wanted to make that correlation. And right now, I want you to just think of it as a correlation, but I did want to get that out there. There is a link that I'm going to try to establish for you, possibly a unicorn or maybe real, between eating, changes in the gut microbiome, and mood disorders, among other types of things. We never used to think of it that way. And in fact, one of the things that we now know is that drugs like Prozac, this popular antidepressant, will affect the gut microbiome and the types of bacteria that grow in your gut. We never used to even consider that. Again, crazy idea that we never thought of that before, but we now do, and we will also be changing the levels of serotonin that result, and this changes the gut microbiome. So we're always finding out new things, and ideas that we never would have thought of before that seem to make no sense, now we're going back and re-examining things. So brand new, amazing field, incredible sort of perspective change that is going on. So great time to be in neuroscience. And it's not just about mood disorders. So let me make that really clear. We now also think that many of the neurodegenerative conditions and developmental conditions such as autism spectrum disorders, Parkinson's disease, Alzheimer's disease, they may actually begin within the gut. And it is not just correlational. It's not just that, oh, they have gut-associated issues. It may actually begin there, and more evidence came out this summer to make stronger and stronger links that, in fact, some forms of these neurodegenerative conditions, like Parkinson's, actually begin in the gut. And it's, again, an incredible shift in the focus that we have today versus what we had a few years ago. So I introduced this idea of this microbiome, particularly within the gut. And it used to be that all bacteria were considered to be bad. So you had like one type of bacteria, and this one type of bacterial infection would then cause a particular type of pathology or disease. So think about staphylococcus and staphylococci, or streptococcus in better terms. And it turns out that we need to rethink that idea in terms of microbiology. So the microbiota has all of the different bacterial cells, viruses. Viruses are also going to be a big part of our course. Various eukaryotic organisms like fungi that can grow either on the body, inside the body, within the gut. And we have a lot of these. We have like a quadrillion viruses that we think grow on our body all the time. And we think that all of these combined, this microbiota, all of these different organisms play a role in keeping us healthy, not just within disease. Okay, so it's been a really good, strong shift over the last 10 years within microbiology as well. This concept is an important one. And again, you don't need to memorize everything that's on this particular slide, but just to let you know that as you do these different types of things, you change your lifestyle and you start exercising, or you change your diet like Gwyneth Paltrow and I did, or you change your hygienic types of behavior, you're going to alter the microbiome. Antibiotics are not the only way to do this. That Prozac that I showed you earlier is also changing the gut microbiome. And we know that by changing the gut microbiome, it can lead to chronic forms of inflammation. We can also cause metabolic dysfunction. So remember, I talked to you about my own weight gain and the fact that I can't actually get weight off anymore. So it's causing metabolic dysfunction. And these involve the gut microbiome, and we'll be talking about that. We know that there's a genetic component to all of this. It's not just what you have already growing inside of your gut, but it's also the genetics of your gut as well. And we will talk about altering these gut microbiomes or intestinal microbiota that might lead to various diseases and degenerative processes, whether they're related to mental health or to neurodegeneration. So again, exciting times ahead, folks. And again, if at any point in time you need to take a break, because I've just been talking nonstop, it's that exciting to me, feel free, and hopefully you can come back at any point afterwards. We're almost there though, so just bear with me if you can for about another 3 or 4 minutes. So this idea of this dysbiosis, which is an imbalance in the normal microbiome that we have normally. If you're healthy right now, you are not in dysbiosis. But if you have an imbalance because you've stopped exercising, you're taking different drugs, then you're going to change what gut microbiota you have, and this will result in dysbiosis. And we know that within adults, the bacteria, viruses, and the prokaryotic populations tends to be very, very stable. As long as everything that you're doing and as long as everything that you are doing keeps you healthy, then these all stay very, very stable. However, in conditions where you're changing the diet, if you have a disease, you're getting different types of drugs, or you're changing the environment, you're getting more stressed out, there's an environmental change that's going on, this can actually change your gut microbiome. And within the virome, the viruses and all of their genetic components, they're also shown to be stable over time. So it's not just bacteria, it's also viruses, eukaryotes, etc. Diets are a big way to change all of this, and it will be a big focus of what we talk about starting next week. So the slides here on the left, I had actually asked in class. You can probably guess. You don't need to memorize these. It's just to have a discussion about the different types and the different ways that we name the different types of bacteria. So these cocci, a singular is coccus, means that we have a round-shaped bacteria under a microscope, and we can stain them using what is known as a Gram stain. So Gram-negative means that they don't stain for the different dyes that we use in the Gram stain, and Gram-positive is a positively stained bacteria. And after staining, we can see if they're round or if they're elongated and elliptical in shape, like this bacillus, and these bacillus, singular for bacilli, are another shape that's very common to bacteria. And we've already seen these spirochetes earlier. This is that Borrelia that I showed you that's associated with Lyme disease. So we have a lot of the bacteria within our gut. Most of these are anaerobic, they don't require oxygen, and we have somewhere between 500 and 1,000 different bacterial species. Again, they make up only about 3 or 4 different phyla, even though we have different species of bacteria. And one of the concepts that I also emphasized at this point was that having more diversity of these different species is better for you. The more diversity you have in these different bacterial species, the healthier you are. In every condition that we look at, in neurodegeneration, in autism, everything that affects the brain, there's a reduction in diversity. Parkinson's disease patients have lower levels of bacterial diversity. They have lower numbers of species than someone who is healthy. An unhealthy individual will have like 500. A healthier individual will have closer to 1,000. So I'm hoping that point is a clear one and one that comes across for you. The most abundant phyla, again, the small number of phyla, are especially in mice, bacteroidetes, which are gram-negative rods, and firmicutes, which are gram-positive. We will also see other phyla like the proteobacteria or the Variomicrobia and the actinobacteria as well, but these are the major ones that I've highlighted for you. And again, I asked you not to know the numbers here on the right, but just to let you know, we have over 500 different species. Throughout the gut, we will have well over 10 to the 14th number of microorganisms, including bacteria. And one of the things that we see in the stomach, we have very, very low numbers, about 100 to 1,000 lactobacilli species. So very, very low numbers. The stomach is very acidic, and so most bacteria die off within the stomach. And then as we go into the first parts of the small intestine, like the duodenum and the jejunum, and then the last part, which is the ileum, we will start to increase the diversity. So you're seeing not a lot of diversity, more diversity, where we get streptococci and bacilli, lactobacilli. So you know that these are the elongated rods. These are more like the small round-shaped bacteria. You do not have to memorize all of the types of bacteria here, but the one constant is that we do have these lactobacilli within the large intestine, which we call the colon. And again, there's been a lot of debate about the reasons why we have an appendix, but we know that if you have appendicitis or you remove the appendix, this will also affect your susceptibility of developing neurodegenerative conditions like Parkinson's. And I'll talk about that in the next lecture. Pretty fascinating if you think about it. Hopefully you were in lecture and this is just a review for you, but this is sort of an encapsulation of what we talked about. So these gut microbiota are dominated by very small numbers of these phyla, and the major types of phyla, which I'm just using a generalized term of divisions, are these bacteroidetes and these firmicutes. We know that most of you will have variations and various ratios of these bacteroidetes and their species that are associated with them and firmicutes, and we call this your enterotype. Every one of you has your own unique enterotype. And the dominating bacterial enterotypes have these genera of bacteroids, Prevotella, and Ruminococcus. So these are the most common types of bacterial genera that we have, and you will all have varying ratios. So I may have more bacteroids than some of you will have more Prevotella, but we'll all have these major subtypes together. I'm hoping that's clear, and I'm hoping that you're all good with that particular concept of enterotype, and that these are the major types of bacteria that make up these enterotypes. We're good? I'm hoping that we're good. And I kind of ended with this, that this is an exciting time for us as neurobiologists, because now in neurobiology, we're at the place, and we're best suited to be able to look at how changes in the gut microbiome by either donation of other microbiomes or synthetically creating different microbiomes and transplanting them into mice can lead to changes in behavior or changes in neurodegeneration or changes in development. Very, very cool. Or how different types of diet and genetic background can do the same thing. And again, we're in this amazing place right now. Be excited, stay excited, because it's going to be an incredible ride as we start looking for these different types of unicorns. I don't think this is a unicorn. I'm going to try to convince you that it's not, but ultimately, I want you to be critical of the research and the data that I'm going to be sharing with you. And lastly, just sort of as a takeaway or key takeaways, I wanted to just give you a highlight of all of the changing perspectives in neuroscience, the way that different types of bacteria and viruses stay stable, and that we no longer think of them as being pathogenic, but that they're actually beneficial. And that as long as you're healthy, we have specific types of bacteria that keep us healthy, and these are known as enterotypes, and that the microbiome might actually have an important role in impacting the brain. And we talked about Borrelia, and apologies on the spelling mistake here, Borrelia possibly being pathogenic within the brain and causing things like Alzheimer's, and the fact that we normally have lactobacilli within the gut, and that it colonizes the entire length of the gut, and we need those types of species of bacteria in terms of our gut microbiome. Again, apologies on having to record this a second time. Thank you for allowing me to do this director's cut. Thank you for being an amazing group of students. I hope you've been listening right to the end, because I know we don't tell you often enough how amazing all of you are, and I really do sincerely think all of you are fantastic. I look forward to the rest of the semester with you. Please stay excited. Better times ahead, folks, and hopefully all the technical issues will be sorted out by the beginning of next week. If I don't see you before then, have a wonderful weekend. Have a wonderful week. See you in class next Wednesday. Thanks, folks. I'm going to give you a hand right now.